The cell cycle describes the life cycle of cells, from growing to cell division to create two identical cells, and then back to its normal growth phase. Check out the previous podcast to learn more about the individual phases of the cell cycle. In this podcast, we're going to answer one specific question. How do cells know when to divide? The cell cycle has built-in checkpoints that are going to send signals and check cells for accuracy as they move through the cell cycle. Let's take a look at each of these checkpoints to understand how cells divide throughout the cell cycle. When a cell is growing in G1, it's going to be doing its normal cell function. As cells get to be too large, we know that they have inefficient cell transport in and out, and they would need to divide at this point. This brings us to our first checkpoint. It's called the G1S checkpoint because it's between phases G1 and S in interphase. If at this checkpoint a cell gets a stop signal, it's actually going to exit the cell cycle and move to a phase that we call G0, which is the resting phase of the cell cycle. Some cells, like our nerve cells, are always in the G0 resting phase. They never divide. Other cells, like our liver cells, spend much of their time in G0 until they are signaled by some severe damage to regrow themselves. And our muscle cells, which have multiple nuclei, are often in our G0 resting phase. When you lift weights and your muscles get bigger, you're not actually making more muscle. Your muscle cells themselves are just getting bigger. Other cells, like epithelial cells, cells that cover and line our bodies, are rarely in G0 because they're constantly dividing to make new cells. So, if at the end of G1, at the G1S checkpoint, our cells get a go signal, the cell cycle will continue. The DNA will be copied during the S phase, and then the cytoplasm and organelles will get created in the G2 phase as the cell prepares for mitosis and cytokinesis. However, before it can reach mitosis, it's going to hit another checkpoint. This is called the G2M checkpoint because it's between G2 of interphase and mitosis. Here, this checkpoint is going to determine whether the DNA has been copied correctly. If there are any mistakes in the DNA, two things can happen. One, we can use DNA repair enzymes to fix the mistakes, and the cell can continue on in the cell cycle, or the cell might commit suicide and go through apoptosis to prevent incorrect cells from being duplicated. However, if at this G2M checkpoint the DNA has been copied correctly, the cell is then ready to move on to mitosis. Remember, mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. Between metaphase and telophase, we're going to hit the spindle checkpoint. The spindle checkpoint is going to look for 2 things. Number 1, it's going to look to see if all of the chromosomes have been attached correctly to the spindle. And it's going to determine if the sister chromatids can separate correctly, half to one end of the cell, the other half to the other end of the cell, to create 2 identical cells in the end. If both of these questions, the answer to both these questions are yes, the cell will continue through anaphase, separating the sister chromatids, telophase, as the chromosomes move towards opposite ends of the cell, and finally cytokinesis, so that one cell becomes 2. So here, again, we can see that we have 3 specific checkpoints during the cell cycle that are going to control when and how the cells divide. Let's talk a little bit about the signals that the cells get to divide initially in that G1S checkpoint. These signals are controlled by 2 different molecules. The first are called CDKs, or cyclin-dependent kinases. I like to think of these as cell division kinases. Remember from our cell communication unit, we talked about kinases as special enzymes that are going to activate other proteins by phosphorylating them, adding phosphate groups. So these kinases are going to set off a signaling cascade, phosphorylating different proteins to help to start that cell division process. Every cell has a constant amount of these CDKs that are present in them at a given time. However, usually these CDKs are inactive. The other molecule that is really important are called cyclins. Cyclins are going to combine with the CDKs, cyclin-dependent kinases, to activate them. They're going to attach. And that's why the CDKs are called cyclin dependent. They aren't active until cyclin binds to them. Once the cyclin and the CDK bind, it's now an active molecule. And we now call it a MPF, or mitosis promoting factor. The higher the number of MPFs there are in a cell, the more the cell will divide. So let's take a look at what happens to the concentration of these two different molecules over time. Normally, our cyclins are low in our cell, but as they increase, what will happen is the MPF activity will increase as the cyclins and the CDKs combine. You can see that as the cyclin amount increases and the activity of the MPFs increase, the cell moves through the cell cycle. Once it reaches its peak, we have mitosis and the cell actually divides. As soon as the cell divides, the cyclin's concentration decreases significantly. Without cyclins, those MPFs become inactive and our CDKs and cyclins separate. And that brings us back to the G1 phase of interphase. But as the cyclins increase, the MPF activity also increases. The cell moves through the cell cycle into mitosis. Cyclin levels drop, MPF activity drops, and the cells move back to G1. Again, our cyclins are going to increase and the whole process starts again. So, our cell cycle has 3 specific checkpoints and are controlled by internal molecules to send signals to divide. Any issues with these checkpoints can result in uncontrolled cell division and cancer. I hope that this was helpful. I'll see you guys next time.